The study I presented is sort of the first part of the story. That the second part will be presented later today by Stefan Pfister. And this is that we look at pediatric tumors, and we look for options that we find to do treatment based on genomic alterations in the tumor genomes of these children. And in particular, the focus that I presented here was on pediatric brain tumors. So I talked about novel findings that we have in terms of pathomechanisms that are possibly targeted, findings that there are a few alterations that are presented for which already drugs exist, and what we learned when we treat patients. And one particular aspect was that we also look into the detail of possible hereditary predisposition of these tumors, and we found that in some subgroups of these pediatric brain tumors the hereditary predisposition is much, much higher as expected, and this has immediately consequences for genetic counseling, for the modalities of treatment and so on. This was particular in medulloblastoma, which is the most common highly malignant brain tumor in children. And there is a subgroup that is called Sonic Hedgehog subgroup, because this particular biomedical pathway is activated. And in this subgroup, 1 in 5 children have a hereditary predisposition, and this is basically affecting 6 different genes, some of the well-known ones like TP53, but also other ones. Yeah, it's very different depending on the subgroups that I briefly addressed. There are 4 main subgroups. The first one has a very, very good prognosis and a very good cure rate. Almost everyone is cured. However, unfortunately, it's the smallest subgroup. And there are 2 other subgroups for which I presented today a number of new pathomechanisms that could possibly be targeted. And for those, the 5-year overall survival is not as good, and therefore we hope that we can, by these new targets, offer some new alternatives in the future. So we find very interestingly, we find rearrangement, so that a gene is basically translocated onto a regulatory element, such as an enhancer very often, which comes from a very different gene, from a very different genomic area, and because of this, the gene is regulated in a way that it should not be regulated, and therefore this contributes basically to the malignancy of this particular cell, to the progression and sometimes even dissemination to the cell. So this is a phenomenon that was known years ago from a few lymphomas, but now we also find it in solid tumors. And we find it specifically in some of the subgroups of the medulloblastoma, with some specific genes with specific enhancers. If we now get all the findings together with the guidelines, we have to keep in mind that, for instance, for the Wnt subgroup, there is a very good successful treatment, as I said. But there's a very, very high percentage of a mutation of a particular gene, Wnt-Catenin. If this mutation is not there, then we have this inherited situation with a different gene, APC. So this would be specific guidelines, even though it's a good subgroup, screen for this mutation. If it's not present, go for the other gene and consider counseling. In other cases where we have this, as I just said, enhancer hijacking in group 3 and group 4 tumors, there we need to develop a therapeutic option. So we need to do more preclinical work. That's what we are doing in animal models at the moment, to see whether we can achieve site-specific enhancer inhibition. That's in principle doable, but we have to show it.